Now, with immunology, right? Immunology really speaks to the body's general responses to any type of foreign material that enters to disrupt any kind of homeostasis in order to take over specific somatic cells, right, or in order to propagate within the body, right? Anything that doesn't have genetic markers, right, that are from the body itself, right, will be most times, right, it will be treated as some type of pathogenic material, right? So would have described pathogens before in CSEC, would have known what pathogens are and the five major categories of pathogens, right? I'm not sure if you guys could remind me of what these different category of pathogens are. Just remind me, what are the different pathogens?
You have viruses, bacteria, fungi, um, pro, we can't really pronounce the one
Protozoa.
with
Protozoan.
Mm-hmm.
protozoa. How much that?
That's four.
And parasites. Did I
Parasites.
say?
Lovely. Right? So those are all the five, right? Majority of the five, right? You also have a subset of some materials that are not necessarily alive, but not necessarily viruses either, and these are components, right? Allergens that are types of antigens, right? So we could speak about these different things. Everything should be generally good, right? So we know what pathogens are. What are pathogens? What are pathogens?
Microorganisms that causes disease.
Microorganisms are genetic organisms that cause diseases, and diseases are any type of malfunctioning of um, metabolic mechanisms, right? So anything that's not going well, or anything that any function, right, that is not actually doing what it's supposed to do, that is generally what we describe as diseases, right? So we have different categories that we spoke about, but we're gonna be that we spoke about in CSEC. We're gonna be speaking about infectious diseases specifically, right, and looking at how these pathogenic components, right, would be mediated or be, you know, defended against, right, within the body systems, right? So we're not gonna look at the immune system specifically, right? But we're gonna be looking at certain components of the immune system as well. We're not gonna look at the lymphatic system, but we're gonna mention the fact that lymphatic system has some amount of a role to play here. All right. So first, we're looking at an immune response, and generally, what the immune response describes per se, right, is the defensive mechanism that the body takes towards these foreign substances or antigens, right? So the antigens such as pathogens, toxins, and any type of harmful agents, allergens, etc. Right, so we're going to be looking at these complex mechanisms that the body creates, right, in order to counteract these changes, right, or the presence of these foreign materials. So an antigen specifically, right? So an antigen really just speaks to a substance, right, itself that may be produced by a pathogen or may warrant an immunological response, right? So antigens tend to be some type of glycoprotein or protein or polysaccharide or glycolipid, anything within the cell membrane, right, of cellular pathogens, right? Because you know you have pathogens are not cells, right? So cellular pathogens, right, usually have these different substances, right? We also have different types of projector proteins that are present within viruses, right? I noticed that when I was looking at the syllabus that you don't specifically look at the structure of viruses or anything viviridae in nature, but that's something that we extensively did when I was doing Unit 2 biology, but I just omitted it overall because I guess it's not in the syllabus. So I guess you don't need to know the structure of viruses. Okay, that's weird, but okay. So we have that there. So we have different types of toxins created by the fungi, viruses, bacteria, protozoa, right? These different organisms, right? And these toxins can be generally just allergens, right? Or general antigens, right? Well, I should say specific allergens or general antigens, right? So allergens, well, antigens specifically, right? Antigens specifically have what we call a hapten, right? Which is a small molecule that is not really used for immunological responses or anything like that. That is attached to the larger carrier molecule. And we also have some things that we call epitopes, right? So epitopes are usually known as allergenic determinants, right? So these are the specific parts of the antigen molecule, right? These are specific parts of the antigen molecule, right, that the B cell and the T cell would respond to, that are macrophages, are monocytes, right, would actually refer to, are eosinophils, right, or neutrophils. These are different things that we're gonna take, right, generally are antigen epitopes, right, and use them, right, in order to present, right? So you have linear epitopes, you have conformational epitopes. Linear epitopes are generally just sequences, right, of proteins, right? Generally primary structure. They tend to be linear, and those generally appear within simple cellular cytoplasmic membranes, right? While the conformational epitopes are usually three-dimensional structures, tertiary structures of proteins, right? That are found within more complex antigen, pathogens rather, right? So we're looking at generally the structure of the antigens, and these epitopes and those type of stuff. They're going to be the things that are going to trigger our major histocompatibility complexes that we're going to look at within the cell-mediated immune response. So my bad. So that is generally there. So we have haptens, right, and the carrier molecule, right? We have a complete antigen like that, and it's usually the different projector proteins on the surface of the antigen, right? And well, bearing different epitopes that we're going to be looking at with and how it affects or activates things like our mast cells, our NK, well, not our NK cells, but our cytotoxic T cells, right? And stuff like that. We're going to be looking at how they activate, right? Just another thing, antibodies. We know that immunoglobulins are generally what we talk about within the humoral response, right? So these are different types of small proteins, right? Usually Y-shaped, right? Tiny proteins that trigger the neutralization or the destruction of different cells or different pathogens, right? And these are usually produced by different immune cells. So usually the plasma cell, I'm gonna talk about it, right? So the general structure, we generally have two heavy chains, two light chains, right? Linked by disulfide bonds, and then and then it forms this Y-shaped structure, right? So the heavy chains specifically, right? These are generally identical, right? So the two chains will tend to be identical. So we're gonna have two identical heavy chains, right? That are large polypeptide chains, right? And they determine the class of the antibody, right? So there are different classes to antibodies, but we're not going to be looking at it too in depth. I don't want to go through it because, yet again, this is something I went through before, but it's not on the syllabus, so I guess I'm not going to go into it, right? The heavy chains have a constant region, right? The FC region that determines the class of the antibody, right? And the effector function. So it determines the class, right? And the class really is what we group them into when we look at different functions of these antibodies. All right. Everything's good so far? All right. For the light chains, now each antibody would have two identical light chains. So the chains are identical to each other. Two heavy chains are the same. Two light chains are the same, right? We generally have two types of light chains, right? The kappa and the lambda light chains, right? But we do have both. So either we have two kappa or we have two lambda. But yet again, I just put one line there because it's another thing that I went through meticulously but it's not on the syllabus. But the light chains also have constant regions and variable regions on it, right? So looking at the variable region now, the variable regions, right, of both the heavy chain and the light chains, right, have antigen binding receptors, right, at the tips, right? So they have binding sites, right, at the tips of the Y-shaped structures that we can see. We're gonna look at the structures, right? They're generally sequence of amino acids, right? And these amino acids would have different R groups that create different types of structures that will actually interact and go through conformational changes when it binds to a specific protein, right? And the constant regions themselves, right, they don't change, right? So when we're looking at the different components of the immune system, right, such as complement proteins and phagocytic cells, right, the constant regions are present there, right? So they're involved in components and complements, right? And we're gonna look at complements when we go through phagocytosis, right? And looking at how cytotaxis occurs, right? So the region determines the antibody's isotype and mediate effector, right? So it's generally the constant regions that determine the class, right? And the variable regions determine what receptors it will bind to, what effects it will what specific receptors it will bind to. That's really it, right? It's like enzymes, lock and key, right? It's specificity, right? And we have the disulfide bonds connecting these chains together. So this is generally what we have, right? So we have the heavy chains, right? And we have the light chains, and we have these hinge regions of disulfide bonds that hold them together, right? And they're gonna have the antigen binding sites, and then that's where the antigen will come into. All right, that's really, really simple, isn't it? Everything's fine here? All right. So notice how that's generally why we call it Y-shaped, because of how it really looks like this. So it's Y-shaped. All right. We're gonna be looking at its function, especially when we look at monoclonal antibodies a tad bit later. All right. Now the cell maturation. Before I go into the entire response, I want to look at the cell maturation first. All right. So what I notice is that module 3's immunology looks a little bit weird. So I'm gonna input things that I think need to be inputted and switch around the objectives, right? Because some of them don't make sense in their current position. So I'm gonna put this before everything else, and then phagocytosis before cell mediated and stuff like that before. So it makes one nice cohesive topic, right? So the cell maturation now, we know that we have T cells and B cells. Those are the general cells that we look at, right? We have a way more cells, right? But we're just gonna look at this for the applications of biology, right? Simple unit 2 biology, okay? So the origins, right? We know that both of them have the same origin within the bone marrow, right? So generally cells are created within the bone marrow there, right? You could look at that there, right? And for the T cells and the B cells, we have the different locations of maturation. So the T cells with the thymus, the B cells with the bone marrow, right? We have the selection processes, right? So the T cells have positive and negative selection in the thymus, and the B cells only have negative selection in the bone marrow, right? We won't go into that too much, right? But we have different maturation steps. So we just need to know where they originate, where they mature, right? It's generally there. Okay. So where do they migrate after, right? After entire differentiation, stuff like that, where do they actually go? So the peripheral lymphoid organs, right, after the thymic maturation, right? So the T cells are generally going to be within that region, and the B cells would generally be within they will actually go toward the lymphoid organs as well, right? So when we're looking at lymph nodes and stuff like that, the cells are going to be going towards those regions after maturation, right? So could look at this. So the B cell maturation, T cell maturation, and we also have macrophage development here. Generally, right, they tend to occur within the same place, right? So the T cells will go to the thymus to mature. The B cells will stay in the bone marrow to mature. The monocytes, however, right, the macrophages are not actual macrophages yet, just yet, right? Because they will just be monocytes, right? As we described a little bit within the circulatory system, right? They would be present as monocytes, and then differentiation would occur when there's some type of you know growth factors being presented to the cell within tissues, right? And when there's some amount of response, right? So when there's some amount of antigens present, the monocytes will generally mature, right, into macrophages, right? So the humoral response, now we know that the humoral response is primarily mediated by the B cells and involves the production of our antibodies, our immunoglobulins, right? And we call it humoral because they circulate within the humors, right? Or what we call the body fluids. Those are termed as the humors, right? So both blood and lymph are humors. So because we're having this response and the antibodies flowing through the blood and through the lymph, right? It is called the humoral response, right? So the first thing we generally look at within humoral response is antigen recognition. So the B cells have these different types of sites, right? We're going to be looking at them. So each B cell has a unique B cell receptor, right? A BCR on its surface, right? And these unique B cell receptors will bind to the epitopes of different antigens, right? And when an antigen enters the body, it may encounter the B cell receptor, right? And then, well, of a specific B cell, right? So the B cells have some amount of specificity to the antigen. So these are antigen specific processes, right? So when we look at the humoral response and the cell mediated response, these are antigen specific responses, right? So this is just how to look at the different epitopes and the fact that the epitopes are specific. So when we have B cell receptors, these are antibodies at this point, right? But when we have B cell receptors, right, the B cell receptors will generally bind to these specific epitopes, right? So they have some amount of specificity to it, right? So there are different receptors for different antigens, right? Here, we're just looking at this. We're looking at the extracellular antigens. They're going to bind to our B cell receptors, right? Specifically, right? And then the B cell itself will actually carry this entire complex itself, right, and bind it to a T helper cell, which will release cytokines that helps within the maturation of the B cell, right, into different memory cells, into different helper cells, and into different plasma cells, right? So what we're discussing within the humoral response is really looking at the HLA class 2, right, fragment specifically, right, after the extracellular antigens are binded to the B cell itself, right? It's going to take those fragments and it's going to create what we call an HLA class 2, right? And it will present that antigen on its surface, and then that a T cell, right, a T helper cell in this case, right, will actually bind to that complex, right, and start to produce cytokines, right, and these cytokines will help within the clonal maturation, right, of different B cells. We're gonna have the multiplication, clonal expansion, right, and the B cells will continue to divide and produce plasma cells for the most part, right? We have different cells being produced also, but right now we're focusing on the plasma cells. And the plasma cells would be the cells that primarily create all these antibodies, all right? So we're going to be looking at that. So we're going to have clonal expansion, right? And all the progeny become antibody producing plasma cells, or majority rather of the progeny, right, will become antibody producing cells, all right? So the antibodies themselves will go through. So what we're looking at here is activation specifically. How does that really occur? After the HLR complex is created and presented onto the surface of the B cell here. So looking at the foreign pathogen coming in. Let me get this. The foreign pathogen coming in binds, right, and allows for the presentation of this complex here. Right, this is a bit different. This is generally the gonna be happening within the this is similar to what's happening within the cell mediated response, right? But we're gonna get there. So we have the antigen being presented there, a piece of the antigen being presented here, right, within this complex, right, and the T helper cell, right, will actually bind, right, and produce cytokines, right? Right, so cytokines, right, would actually go and help within the proliferation and clonal expansion of the B cell, right? So after we have that, we're gonna have the proliferation and differentiation here. We're gonna have the entire complex, right? We're gonna have the T cell and the B cell receptors, right, combining, and we're gonna have the production of the cytokines, right? That will allow for the clonal expansion of the B cells into memory B cells and long-living plasma cells, right? So some of the progeny would actually become plasma cells, some would become memory cells. We're going to be looking at the key roles of the memory cells in a bit, right? But the B cells are generally what the plasma B cells rather are going to be generally what we're looking at within the humoral response, as they would produce majority of the antibodies that are specific to the antigen detected. So the antigen that is detected, right, specifically, will cause the B cells to produce an antibody specific to the pathogen that presents that same antigen, right? So what the antibodies would do is going to identify the specific pathogen, right? And there's a lot of specificity to this. So this helps within specific instances where a pathogen, right, is completely remembered, or a pathogen has entered the body before. This will assist within a secondary response, right? Because it's so specific. But there are other responses that are not so specific, right? And that are general in and of itself. So we're generally looking at this, right? We have the we have a general B cell, right? So we have the T cell independent activation. What we're talking about, right, is the B cell will actually bind to the antigen, right? Break it up in parts. Piece of the protein will be presented on a complex, right? Generally called the HLR complex or the MHC complex, right? We're gonna look at that a little bit more in depth, the formation of the complex in phagocytosis. What it will do is present that complex on its surface. A T cell will come and bind to that complex, and the T cell, the helper T cell, will now produce cytokines, right? That will actually help within the clonal expansion, proliferation, and differentiation of the B cell into its plasma B cells and the memory B cells. And we know that the memory B cells will be there for the secondary response, right? And the plasma B cells will be there for the production of antibodies. All right. So antibody production here, this is really general. All right. So when the plasma cells are highly specialized antibody producing cells, right? So they produce and secrete antibodies at a very high rate. And when these antibodies are produced, they travel through the humors of the body, the blood and the lymph, right? Where they will encounter and bind to target antigen, right? So that's generally what the humoral response is all about. Right? That's generally it. Right? So the neutralization, elimination, what the immunoglobulins will do, right, is actually bind to the different pathogens, right, mark them and neutralize them, right? So you're gonna have neutralization. So the antibodies can neutralize the pathogens by binding to the critical sites of the pathogen and blocking its ability to infect other cells, right? So that's generally the neutralization, right? We have opsonization, right? What we're basically talking about, right, is the fact that antibodies can mark pathogens for destruction by binding to the surfaces and making it easier for phagocytes to locate them. So that's one thing, right? And we have complement activation as well. It can activate the complement system within the immune response, right? And we're gonna look at what complements do. And they also have antibody dependent cellular cytotoxicity, right? Where antibodies can recruit natural killer cells to bind to the FC complexes, right, of the antibodies, right? Generally bind to the antibodies themselves, right, and cause the natural killer cells to destroy or cause apoptosis to the target cells, right? So the focus of the lymphocytes specifically, they will bind to the lymphocytes will bind to a specific antigen, present it onto their surface. T helper cells will actually bind to that complex, right, that is presented on the surface, produce cytokines. Cytokines help within the proliferation and maturation and differentiation of these different B cells. So we're gonna have plasma B cells, memory B cells. Memory B cells are stored. Plasma B cells are gonna be producing our antibodies. What our antibodies will do is either neutralize the pathogen, right? Mark the pathogen for phagocytes to be attracted or for natural killer cells to be attracted. Generally, what the entire thing on the antibody does, right? It allows for the easier destruction of the pathogen, right? And that's really important. So they defend against any pathogen by either neutralizing their toxins, right, or making them clump up, right? They can actually make them clump up together, right, and actually allow for phagocytes to easily engulf them, or they can act as a marker for natural killer cells to destroy them, right? So that's generally what they're going to be doing, right? That's generally the humoral response, and this is a good depiction of the humoral response generally. Okay? So it's really simple. Antigen recognition, antigen presentation, right? T cell combination, proliferation of memory and plasma cells. Right? Plasma cells would produce the immunoglobulins. And that is really it for the humoral response. All right.